Hello, good afternoon, everyone. Welcome back to the second half of the course. I hope you had a good engagement week and you were able to had some uh, rest and all recharge for the second half of the semester. If you didn't know this, the second half of the biology course is kind of a standalone from the first half. All the stuff that you were tested on will no longer show up on the evaluations. So we're kind of starting brand new, if you would. And the second half is pretty much the same structure as the first half. There will be five lectures and two quizzes, quiz three and quiz four, 10% each. There's going to be another lab as well as the final test, which is 20%. Um, and we're going to be starting with lecture six today on the topic of cellular reproduction. So let's get right into it. Before we go into the actual cell division, because cell division involves the passing on of genetic information in the form of DNA, I'm going to go through a little bit of brief history related to how they discover DNA as the molecule for transmitting hereditary information. Now, this picture, what you're looking at is actually really powerful magnification, a microscope, that are looking at viruses. Okay, so these little green blobby stuff, those are viruses. And what they are infecting here is actually a bacterium. Okay, so you can see the viruses are injecting something into the bacterium, and that something is the genetic material of the virus. We know now that the genetic material is DNA, but before people didn't know. Okay, they are mystified. What is that stuff that's being injected? Is it DNA? Is it protein? What is it? So we're going to be going through a series of experiments. We're going to be taking notes on these experiments that will tell you how they've discovered DNA as the hereditary molecule. Okay. Now, these experiments, I'm going to warn you ahead of time. They are, could be complicated. So please stop me if I am not making sense and if you need me to clarify or elaborate on some of these stuff. All right. First question. What are the hereditary materials made up of? There were two camps of scientists, if you would. One camp believed that it was proteins that's responsible for being the hereditary material, because as you know, proteins are made up of 20 amino acids, whereas DNA are only made up of four nucleotides. So intuitively, you would think you are able to code more information with 20 building blocks as opposed to just four building blocks, right? So people believe it is the protein that's being the hereditary material. But then the DNA is also a very complicated molecules that has very elegant structures, and some people believe that that would be the hereditary material instead. And so people back and forth didn't really know. There was no experiment to show it one way or the other until 1952, where a pair of scientists by the name of Hershey and Chase, they conducted a set of experiments that would definitively show DNA is indeed the hereditary materials and not proteins. So we're going to go ahead and take a note on the Hershey and Chase experiment. And like I said, you're going to need two or three colors for this note. Okay. Now I will try to write slower. That might improve my writing a little bit. It's just really hard to write on the screen. Okay. But if you can't read something, just ask me. Okay. I will read it to you or explain it. I'm not going to get offended. Don't worry. So this is the Hershey and Chase experiment. All right. What's the point? Okay, the purpose. The purpose of their experiment was to determine whether protein, protein or DNA is the hereditary material. Okay. What gets passed on from one generation to the next generation? Okay. That's what they're trying to find out. And so they studied this problem using a type of virus called the bacteriophage. Okay. So the bacteriophage, as I show you on the picture with those green little blob things, they look like this. This is a bacteriophage. They infect bacteria. They are harmless to humans. A bacteriophage. And the bacteriophage comes in two parts, if you would. Okay. It has a shell. I guess I'll label the shell red. It has a shell that's made up of protein coat. And then on the inside, they have genetic material, DNA core. Now, they didn't know back then, like what's on the outside and what's on the inside. Okay, we have the benefit of hindsight, right? So we know now the protein is on the outside and the DNA core is on the inside. But they had an understanding of how viruses replicate. Okay, so a quick lesson for you. This would be a host, e.g., a bacterium. A host is the cell that is about to get infected. When you get infected by a cold virus or COVID virus, whatever, they basically would attach to your cells and then they would inject their genetic material into those cells and they would turn your cells into virus factories. So basically, you would have this is the virus lands on it, okay, and then they inject something, injects genetic material, which we know is DNA now, but they didn't know. That's the whole point of what I'm trying to tell you. And then what happens is the virus will turn the cell into virus factories, and eventually the cell, in this case, the bacterium in our body, it would be our cells, would burst open, and then new viruses would come out, and then they would go infect more cells. Okay, so that's basically the cycle of how viruses infect. So they understood that, they knew that. They just don't know what went inside. Okay? So in their experiment, they prepared two groups, two groups of bacteriophage. Two groups of bacteriophage. Group number one, they grew in radioactive, radioactive sulfur. If you remember from your chemistry class, sulfur has a mass number of 32, but these are radioactive sulfur, so they are sulfur 35. Okay? Don't get too confused with like radioactive. Like what does that mean? All that means is you can track it. Right? If something is radioactive, then you can detect where that substance is. So if you grow the bacteriophage in radioactive sulfur, do you think the DNA will be radioactive, or do you think the protein will be radioactive? Which of them contains sulfur? The DNA or the protein? That's right. Protein is going to be radioactive because proteins, as you know, contain traces of sulfur, right? So in this case, this batch of uh, viruses, this batch of bacteriophage is going to have a radioactive coat. The DNA will be inside and it would not be radioactive. There is a different batch, and this time they grew in radioactive. What should we use? If we want to label the DNA and not the protein, what element should we make radioactive? What's something that you find in DNA but not protein? What element? Phosphorus. Okay. Phosphorus is only found in DNA but uh, not in the proteins. And so they use radioactive phosphorus 32. Okay. And this time, the viruses would have a normal protein shell, protein coat, but the DNA inside will be radioactive. Okay. Are we together so far? All right. I'm going to tell you something else here before we move on. So, as you can imagine, the next thing they'll do is they're going to infect the bacteria with these viruses. And what happens is, after they infect the bacteria, they are going to put it into, um, they actually use a blender because they didn't have sophisticated instrument back then. But the point is this, okay? They're going to spin the mixture at really, really high speed. And when you do that, you can actually separate things according to their mass. So things that are heavier is going to get to the bottom, and things that are lighter is going to go on the top. So over here, this is a test tube. And when you spin down the mixture that contains the viruses and the bacteria, what you will end up getting is you will get a top layer and a bottom layer. Okay. The viruses are lighter compared to the bacteria. So at the top here, it will contain parts of the virus that did not enter the bacteria. Okay, remember, they're going to inject something, and then part of the virus is going to stay outside. And when you spin it at really, really high speed, the virus is detached from the bacteria, and it's going to be at this top portion of the mixture when you spin it down. On the other hand, the heavier stuff, this is where the bacteria is going to be. Bacteria plus whatever was injected by the virus. Okay? So that's the setup that you have to understand. All right, let's take a look at the results. So this is group one with this guy. That's radioactive on the outside, and then no radioactivity for the inside. And when you spin down the mixture after the infection, do you expect to detect the radioactivity at the top portion or the bottom portion? Based on everything that we've discussed, do you think the radioactivity from the protein shell will be detected at the top portion or the bottom portion? Okay. Well, let's recap a little bit. The outer portion is the protein, which we know does not go into the bacteria, right? And then the core is where you will have the DNA, and we know now that's the stuff that went in. So over here, if you only label the protein with the sulfur 35, the protein is not going to go in to the bacteria, and they're going to stay outside, and that's the lighter part, and you will find the radioactivity at the top portion. Okay, radioactive. And then the bottom will have no radioactivity. The DNA went in, but it didn't light up the bacteria, so to speak. So what does it mean based on this result? Well, based on this result, it means protein did not enter bacteria. However, this does not prove that the DNA went in. In this particular setup, there's no way to track where the DNA are. Right? We can only show that the protein did not enter the bacteria, but we don't know where the DNA is, which is why they need to repeat the experiment with the second group of bacteriophage. The second group has radioactive DNA. And after infection, and they spin down the mixture in the test tube, okay, this time the radioactivity are only detected at the bottom. So what does it mean? Well, remember, the DNA is radioactive, so it shows that the DNA went into bacteria. Okay, entered into bacteria. So collectively, if you take these results and put it together, what does it mean? Conclusion: DNA, not protein, is the hereditary material. Okay. You cannot just look at the result of number two alone or number one alone. If you look at number two alone, it shows that the DNA went in, but you cannot show that's the only thing that went in. Like maybe the protein went in too. I wouldn't know unless I did the other experiment as well. Right? If I just have the experiment number one, then I know the protein didn't go in, but it doesn't mean the DNA went in, right? That's why you need to have both of these results to be sure. Any question before we move on to more complicated stuff? All right. Now I actually have something to tell you guys. Last week we had a meeting in the department, and we've decided that we have to make some changes to how assessments are being done online. So just like your math class, effective immediately, all the assessments for my class for the biology, and you will hear this from your chemistry teacher as well. Okay, all the assessment done online now will require you to turn on your camera, and notes, electronics, and other help sheets, all that kind of stuff will no longer be allowed during the test. So please be prepared for that, and you might have to adjust how you prepare for the assessments moving forward. Okay. Um, does anyone have any question about that? Okay. So I'll make an announcement to clarify that. Basically, no more notes and stuff like that, right? You have to memorize the stuff and do it accordingly. And later on, quiz four, which I will again mention it closer to the actual quiz day, you will have to solve problems And in math, I know you guys have to submit like rough work and stuff like that, right? Like to actually show your work. So you will be required to do that for quiz 4 as well, because we will be solving quite a bit of problems that have steps and things like that. So just be mindful of that, okay? All right, so that was Hershey and Chase experiment. This is what I was trying to show you with the protein code and the DNA core. And then you can see in the experiment, the protein stayed outside, but then the DNA went in. It's exactly what we wrote down. You can look at the pictures, and try to understand it a little bit more. All right. Now that scientists knew it was DNA that is responsible for being the hereditary material, then there is this race to try to understand and figure out the exact structure of DNA. And this was done by, several scientists, but most notably, um, it was Rosalind Franklin and later on Watson and Crick. So for Rosalind Franklin, she was a scientist who did a lot of work with X-ray. And using X-ray and crystallography, she was able to deduce that DNA has a double helix. But before she can finish her work, she actually died of cancer, probably because of all the X-ray exposure that she had in her experiments. I mean, back then the safety precaution was not, 100 as good as nowadays. So possibly that's where she got exposed to too much X-ray. And in 1953, Watson and Crick continued with her work and basically built upon her data and come up with the first 3D models of DNA that is actually accurate. Okay, and they subsequently win the Nobel Prize for doing that. And some people say it should have been Rosalind who also would share the Nobel Prize, but at the time they got acknowledged instead. Okay, there is a question. Does that mean quiz and tests will be done during class times? Quizzes will still be able to do throughout the day, from 6 a.m. to 11:59 p.m. That test will probably be done during class time. But don't worry about the quick test now. You only have one more test, and that's going to be happening at the very end of the course. We'll talk more about it later. But for the quizzes, it's still going to be the whole window for you. All right. Okay, moving on. Next, we have Messelson and Stahl, and they are important scientists who um, did experiments to figure out how DNA are being copied when they go from one generation to the next generation. Now, just to recap a little bit for you, right? We need to make more cells as we grow, right? To replace lost or damaged cells. And we all began with a single cell, but now we have trillions of cells. And so here we have the original cell. This is called the parent cell. And just for simplicity, it has two chromosomes. Okay. And for us, we of course have 46 chromosomes. And then what happens is the cell will copy all of its genetic material. So instead of one blue and one purple, now it has two blue and two purple chromosomes. And then when the cell divides, these are called daughter cells. And each daughter cell is going to also have two chromosomes. Okay, two chromosomes. So much of Messelson and Stahl's experiment has to do with figuring out how the DNA is being copied to create extra ones to be passed on to the next generation. Okay? And of course, this copying has to be a faithful copy of the original. You don't want to be changing the nucleotides, and if you did, it's going to result in mutation. Okay, mutation. Now let me show you the three proposed model of DNA replication before I show you Messelson experiment. And this you would also need colored. Okay, try to follow the exact color so you don't get confused because the color I'm using is going to match the diagrams I'm going to show you. So please use red and blue as I've used them. Okay. So let's say we have A, C, T, T, G, C, A. Okay. And because DNA strands, they are complementary. If you know one half, you will be able to come up with the other half, right? So in this case, should I ask you guys, what does A pair with? What nucleotide? Hmm. A pairs with T and C pairs with G. Very good. So using this base pairing rule, I can essentially figure out the missing half. Okay, so this is going to be the parent DNA in the parent cell. Okay, parent is not like your parents necessarily. Parent is like the original cell that is about to divide. Now, there are three models of DNA replication. Okay, the first model is called conservative model. And the conservative model goes like this. Okay, you take your original DNA and then you literally make a copy of it. Okay, and then I will use a different color to represent the new DNA. Okay, so that's conservative model. Okay, one strand is going to be made up of old material, the original material, and then you're going to have a strand that is made up of entirely new material. These nucleotides may be just like floating around in the cytoplasm and you're able to use them, okay? So that's called conservative model right there. Now, the second model that was proposed is called semi-conservative. Semi-conservative model. And it goes something like this. Just watch first and then you can copy after. In the semi-conservative model, what happens is you will take apart the original DNA because it's double-stranded, and using the original half, you will use the base pairing rule to build the missing half using new nucleotide. Okay? Like this. So in the end, I have two strands, two DNA molecules. Half is made up of original nucleotides, the red ones, and then the other half is made up of new nucleotides, the blue ones. So it's semi-conservative. You conserve half of the molecule, the other half is new stuff. Okay, just let me know if you don't understand. I can explain again. And then we have the third model. Okay, the third model is called dispersive. Dispersive model. Okay, and it looks something like this. Right? This one is hard to draw. Okay? But basically, you will have you will have bits and pieces of old and new here and there. Okay, it's still, kind of half new, half old, but the old stuff and the new stuff are not half and half on the strand, okay? They are kind of mix match patchwork of new and old like this. And so on the other one, it will look like this. Yeah, I hope this makes sense to you. But in any of these cases, you started with one DNA molecule and you end up having two. Right? The not the argument, but then the situation is people don't know how it happens. Like, is it completely old and completely new? Is it half new, half old? Is it half new, half old, but kind of all over the place, right? That's the thing they were trying to figure out. Okay. And Messelson and Stahl, those two people that I'm about to show you their experiment, is going to figure out which one is the correct model. Now, I made a picture here to help you understand. It took me half an hour to make this. So original copy is like this teddy bear here. And over here, it's like taking the teddy bear and then you photocopy it. Do you think this is conservative, semi-conservative, or dispersive? What are your thoughts?
Conservative.
Yes, that is conservative. Thank you. I was starting to get worried for a second. Okay, that's conservative because it's half new, half old, half new, right? Okay. And this one here, it's like you take the photocopy, but then shred it in half, and then you stitch it with the copy itself. And I hope you understand this is semi-conservative. And finally, it's kind of here and there. It's not restricted to half of the page, so to speak. So this is dispersive. All right. If you're good with these meanings, uh, then I'm gonna go and explain Meselson and Stahl experiment for you. Okay. Like I said, just stop me if you need me to explain. Here we go. Okay, so this is Meselson and Stahl experiment. The other two people, they were Hershey, like the chocolate, Hershey and Chase. And what's the purpose here? Purpose. Purpose of Meselson and Stahl experiment is to determine the model of DNA replication. Is it conservative, semi-conservative, or dispersive? Okay. So what did they do? They do. Okay, so this is what they do. E. coli, as in the bacteria, E. coli were grown in N15. Okay, I'm going to use red for this one. N15 culture for many generations. Okay, do you guys know the mass number for nitrogen? Anybody know that from chemistry? You should know. Or with the periodic table or something. Anyone? Did you guys learn that yet to calculate the molar mass and stuff? Or I'm totally off here. I haven't teach chemistry for like almost a decade, so I don't really know. Have you guys learned molar mass yet? Does that mean anything to you when I say nitrogen 15, nitrogen 14?
15 mass number.
Yeah, the mass number. Yeah, yeah. So, anyways,
Mass.
14 is the normal one, okay? If you look it up on the periodic table, it's 14. So these are 15, which is heavier. Okay, they're not radioactive, but they're heavier. Right? And a culture, if you don't know what culture is basically like a dish and it has nutrients and everything that a bacteria needs to grow. And in this culture, it's full of heavy nitrogen, nitrogen 15. So if you grow the E. coli in N15 for many generations, then all of its DNA will now be heavier because every time it replicates, every time it makes new DNA, it's going to be using the N15 and it will make the DNA heavier. Okay. All right. Then this is what happens. I should probably write it down for you. E. coli were grown in N15 culture for many generations. This ensured all their DNA are heavy. All right. And this is what happens next. The E. coli, the ones that have been growing in N15 for some time, the E. coli were transferred to N14. Please use the color to help you make sense of it. Okay. Transfer to N14 to grow for one generation. And their DNA were extracted and separated by weight. So before we move on, let's try to understand the possible outcome. So again, we have our test tube right here. See if I can draw it a little bit better. And there is the test tube. Now, in this test tube, it's filled with kind of like a gel, okay? So um, it's not like, if I put something in and it will fall right to the bottom. The gel is gonna basically separate the content based on how heavy it is. And you have to spin it down. You have to use a centrifuge to separate the content. Okay. So we have different kinds of DNA now. We have DNA that are made up of heavy nitrogens, and we have DNA that are made up of N14, right? So which one's gonna sink to the bottom? The N15 or the N14? Now that's a rhetorical question, right? I hope you understand because 15 is heavier, it's gonna be down here. Okay? This is the DNA that's going to be made up of N15 only. And if the DNA is made up of N14, then it's gonna be at the top, right? N14 only, because it's lighter. But there is a third possibility. Can anyone tell me what the third possibility is?
Mix N14 and N15 mix.
Yeah, exactly, right? Okay, so you might have a band in the middle. It's not as heavy as the 15 only, but not as light as the 14 only. This is going to be in the middle. It's going to be some N15 and some N14. All right. If you look at our models, this would be a mix, and this would also be a mix. Right? It's some red and some blue, somewhere in the middle. Okay.
I
Now
have one
yeah,
clarification.
please.
The N15 is transferred to N14, the E. coli.
Yes, so they grew the E. coli in M15 for a very long time first.
Yeah.
So all the DNA would be N15,
And
all of them would be pink, so to speak, okay? And then they transfer the E. coli to the N14 for one generation,
yeah.
and then they will extract the
Yeah.
DNA. Yeah. Everyone else understand this thing over here? The rest of the note is going to be based on the understanding of this. The 14, 15, 14, and 15. Everybody good with this? All right. So what's happening? Well, the E. coli that is from the N15, it will look like this, right? Make sure you use the same color as me. This is the N15, N15. When I say N15, N15, I mean like one strand is N15, the other strand is also N15. Okay? So I'm gonna call this generation zero. And then what happens is this is transferred to N14. Now, what happens after you transfer to N14? Now, I'm not going to draw it. You have this in the notes, okay? Because I used to draw it in the past and then it's just like a mess for people to follow. So over here, we have the three situations, okay? We have the conservative over here, and conservative. Sorry, this is semi-conservative. Semi-conservative. All right, this here in the middle is conservative. And over here on the right, it is dispersive, right? So this is generation one. Generation one. So what happens? Well, they allow the E. coli to grow for one generation and then the DNA were extracted and separated by weight. So looking at this picture, these are all the DNA from the first generation. And if you spin it down on the test tube that I was explaining to you, where do you expect to find the band? I'm gonna uh, launch a poll for you, and you're gonna tell me. Okay, where are you gonna find the band? A would be top, B would be middle, and then C would be bottom. Okay, if you think there's more than one answer, you can choose it. Okay. Okay, very good. I have a lot of answers coming in, and it is going to be right in the middle, right? Right in the middle, right? Because half of it is red and half of it is blue, right? Okay, what about this second case here? Right? You collect all the DNA and you spin it down. Where will it be this time? Let me try to launch the poll one more time for you to try. Okay, same choices over here. Okay, very good. We would have a lighter band at the top corresponding to this one, and then there will be a heavier one at the bottom. This would be conservative, right? Comprised of entirely original material and comprised of entirely new material. And finally, for this last one, a dispersive model is a mix of old and new, and therefore you would expect to find one in the middle. Okay, I'll take away that black line here. It's probably confusing people. Okay, there you go. So that's the expected result. What did they see? Okay. The result of the experiment for generation 1 shows a single band in the middle. What does it mean? It means the conservative model is not correct. This means the conservative model is incorrect, because if it was correct, you would have seen two bands. They did not see two bands. They only saw one. However, the data cannot distinguish between semi-conservative and dispersive. Okay, can't tell whether it's this one or this one. They both would look the same. Analysis of generation 2 was needed. Okay, so they need to take this one more generation. So what would it look like? Okay, hold on. Let me copy a little bit more. Now this is in the notes, so you don't really have to draw it. So remember, this is semi-conservative. I'm just going to put semi. This is conservative, and this is dispersive. We already know this is not true. After one generation, we were able to see that it was not true. But if we go to generation 2, right? Listen to me for a second. Right? We're going to separate the red and the blue, and we're going to build the missing half. And remember, only blue nitrogens are available, right? Only N14s are available. So this blue ribbon, you're going to build another blue one, and then you would have the red one combined with the blue one. And if you spin this down on a test tube, you will see a mixed one in the middle, and then you would see the blue one at the bottom. Whereas if the model continues to be dispersive, where the red and the blue are all over the place, then you will continue to see only one band in the middle. Okay. So after the second part of the experiment, this was not observed, and they saw two bands. Okay, two bands were observed for generation 2. Therefore, the correct model for DNA replication is semi-conservative. All right, that's it. I'll give you a moment to catch up, and then we'll continue. Okay, let's do a poll, see how much you remember. This question is about Hershey and Chase experiment. Just look at the notes we took, and you should be able to answer this. Okay, why was radioactive sulfur used? Let's go back to here and take a look. Okay, this is radioactive sulfur, and that allows you to label the coat, and the coat is made up of protein. Okay, so the correct answer here is A, to label the protein coating of the virus. To label the DNA portion of the virus, they would be using phosphorus, radioactive phosphorus. In the experiment, they did not label the bacteria at all. Okay, next question. I'll just do this one with you. If DNA replication follows the conservative model, which of the following would be the expected result from Messelson and Stahl's experiment? Okay, so I'm going to go back here to show you the various models. We have, the question is asking for a conservative model. So after one generation, you would see a top band and a bottom band. And then over here, you're going to see um, also top band and bottom band, right? And so the answer here would be A. Okay. A. All right. Let's move on to the next part, which is mechanisms of DNA um, replication. And first, like much of the things I'm going to show you right now, it actually came from the study of E. coli because they're easy to study and they're easy to grow, and they're simpler than eukaryotes. Okay, and as you will see, there are a lot of similarities between the process of DNA replication in prokaryotes when compared to eukaryotes. And so we will learn it for prokaryotes first, and then we'll extend it to eukaryotes later on. So to replicate the DNA in prokaryotes, the original DNA, the parent DNA, is going to be unwound, and we understand it is semi-conservative now. So each half will be used as a template to create the missing half. Now, this process is actually really, really fast for prokaryotes. The speed is up to 1,000 base per second, so 1,000 ACTGs every second. And in the case of E. coli, it has a total of about 4 million base pair. Right? So it doesn't take too long um, to copy all of its genetic material. It's a little bit slower in eukaryotes. It goes about only 40 base pairs per second because there's a lot of proteins that are stuck to the DNA, and to unwound it takes a little bit more time. And in humans, there are a lot more base pair in total as well, right? Up to 3 billion base pairs. So it does take a little bit longer to copy all of genetic material. And the process involves quite a bit of enzymes and various proteins to help uh, make the process seamless and not create any mistakes. And we're going to be learning some of these proteins. Okay, so they're all listed here, and we will start the note, and then we'll take a break and finish off the note afterwards. Okay, so here we go. I would recommend starting a new page if you can, and uh, just follow along. Okay, so this is DNA replication in prokaryotes. Prokaryotes are like bacteria, right? Bacteria and simpler cells, rather than eukaryotes, which are plant cells, animal cells, um, that sort of stuff. Okay, so let's start small in the corner here. We have two strands of DNA. Okay, two strands of DNA. And DNA are complementary, meaning if you know one side, you can predict the other side. And then we also learned another term to describe the structure of DNA, which is anti-parallel. Right? We learned that in lecture 2. So for anti-parallel, it means the strands are running in opposite direction, but the space between them will always be the same. And we use numbers to um, represent the directions. So on the top strand here, we call it 5 prime to 3 prime. Don't worry too much about why, okay? Like it has to do with the chemical structure. If you really want to know, you can ask me later. But one strand is 5 prime to 3 prime, the other is 3 prime to 5 prime. Just like on the highway, one row is going to be traveling from north to south, the other is traveling from south to north, right? So 5 prime to 3 prime and 3 prime to 5 prime. Now we're going to zoom in on one part of the strand here, and we're going to open it up, and you have to draw this quite big, all right? It's going to look like this. There we go. Okay, so this here is called a replication fork. Replication fork. Okay, you would open up a portion of the DNA. Okay. Maybe I should add a diagram right here. Just include an intermediate steps for you first. This is replication fork. So over here, what's gonna happen is you're gonna create a bubble, and then we are zooming in on one side of the bubble. So this bubble is called a replication bubble. And we're looking at one side of that bubble, which is called a replication fork. Now the number still stays there. This is 5 prime and all the way to 3 prime. And then on the other side is 3 prime to 5 prime. Now this is DNA, so you're gonna have the ACTGs here, and then they're gonna be forming the hydrogen bonds that holds them together. Okay, I'm gonna write a few letters here for us. Now you can make up your own letters if you want. It doesn't really matter, but it's important that you fill it in because I will show you how to build the DNA in a bit. I'm totally just making it up as the letter pops into my head. Now, once you write I mean, you can keep on writing it, but it's just going to be more letters, right? Now, on the other strand, I have to make sure that it is complementary. Okay. So perhaps it's easier if you just copy what I have. There you go. Okay, so that's probably going to take you some time to copy. Why don't we take a break for now, and we'll come back at around 1:35. You can continue to copy whatever you need, and we'll do more afterwards. Okay, see you in a bit. Okay, let's keep going. So hopefully you have the picture with you now, and we're gonna take a look at how the bubble was created in the first place, and then there are other proteins and enzymes that are going to be playing an important role in this process of DNA replication. Okay, so over here we're gonna have an oval which represent an enzyme. This enzyme is called helicase. Right? It's one of the enzymes that are listed uh, in the PowerPoint slide, helicase. So the function of helicase is to unwind the DNA to create the replication bubble. Replication bubble. And this is what we see here, right? So you're going to have a helicase right here on one side. You're going to have another helicase on the other side. It's like a zipper, right? Think of the zipper, the thing that you pull on to unzip the zipper. That's kind of like the helicase. So the helicase will continue to travel into the fork, and it will unzip more of the DNA, and that would allow more base pairs to become available. Now, as you are unzipping the DNA, okay, this part here, I'm just going to continue to draw a little bit more. Remember, the DNA is actually a double helix. Okay, so there are a lot of twists and turns. And what happens is by unzipping the DNA, you actually build up tension in the rest of the double helix. So there is going to be another enzyme that are located further down in the DNA strand. And these enzymes, they are called gyrase. Gyrase. Okay? And the function of the gyrase is to relieve tension that builds up as the DNA is being unwound. Okay, it's being opened, you can say, as that builds up as the DNA is being opened. If we don't have the gyrase, then there will be too much tension building up, and as a result, the molecule might snap, and we don't want that, of course. All right. Don't forget, the base pairs are attracted to each other. Right? The A's and the T's, they have two hydrogen bonds holding them together, and the C's and G's, they have three hydrogen bonds holding them together. So there is a tendency for the two strands to come back together. And of course, we don't want them to come back together because we need to gain access to these nucleotides to build the new missing half, right? The new strands of the DNA. And so we're gonna have additional proteins here. These are called single strand binding proteins. Okay? SSBP for short, and they prevent the DNA strands from coming back together. Okay. Keeps them apart, so they're not going to reconnect. Okay, so those are the three proteins at the top of the list here: helicase, gyrase, and single-strand binding proteins. I'm going to give you a moment to catch up. If you have questions, please ask. Nothing? Okay. Now I am gonna move away from the diagram for a second, and I'll come back to it. But I need to write a bit of a note for you regarding another enzyme. Okay, so this enzyme is called DNA polymerase 3. Okay, so this DNA polymerase 3, this is Roman numeral 3. Okay, DNA polymerase 3 is the main enzyme for building the new DNA strand. Okay. How does it do it? Basically, it uses the parent DNA as a template to create the new strand. Now, 1 second, okay? Take a look here. Basically, don't draw this, just listen, okay? Basically, the enzyme will come, okay? This is the enzyme, it will come, and then it sees that, oh, this is a C, and it knows the base pairing rule. I must put a G here, and I must put a G here, and put a C, and then put an A. And then the enzyme will continue to move, and then you will build the missing half. That's basically what's going to be happening. But don't draw it yet, because there are a couple of problems I need to tell you about. Okay? So, but that's what this means, okay? Uses the parent DNA, which is the original ones, as a template, and it will build upon that template to create the new strand. Okay. So this enzyme has two limitations. Two problems. Okay. First problem, cannot start the process. Cannot start from scratch. Okay. It doesn't know where to start. It doesn't know where to begin. Right? As an analogy to help you understand, it's like my kids. Okay. So they come home with homework. Okay. So like there are many questions here, for example, many questions. Okay. And they never start to do their homework themselves. Okay, maybe these are math questions or something. So I need to sit down with them. And I'm like, okay, let's do this first question. Da, 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 da. This is the second question and the third question. You get it now? You can go on your own. And they're like, yeah, yeah, I got it. I'll do the rest. And then they will be able to finish the worksheet on their own. But someone has to sit down with them to kind of start the process for them, with them. And then they could continue on. And that's what the DNA polymerase 3 is like. It cannot start from scratch. It needs something to initiate the process of DNA replication, and then it can carry on. Right? So I'll tell you what that something is. Number two, the second limitation is that it can only build new DNA in the direction of 5 prime to 3 prime. Can only build in the direction of 5 prime to 3 prime. Just write down first, and I will explain to you what it means. Okay, back to here. So there is an other enzyme, which I'm gonna draw for you now. Okay, I'm gonna run out of color soon. Okay, this enzyme, just gonna put maybe like a square shape here. Okay, this one is called a primase. Primase. Okay, and the function of primase is to lay down a few nucleotides for DNA polymerase 3 to follow. Okay. The primase is like me, the parent, who sit down and do a few questions with the kid before they can do it themselves. Okay, but this primase is here. We're doing this right now. If you're following the list, this primase builds RNA only. So that complicates things a little bit. Right? When you see an A here, what's the corresponding base pair for A in terms of RNA? Does anybody know?
Uracil.
That's right. It's uracil, right? So that's U over here. And then G will go with C, C will go with G, G, A. And it's just going to do a few of them. So this little bit of a starting nucleotide here that consists of RNA. This is called an RNA primer. Okay? It's the uh, nucleotides that are laid down by the primates. Now let's take a closer look at the direction. The black one, okay, it's 3 prime over here and it's 5 prime on the other end, which means on this new one that we're building, since nucleotides are anti-parallel, DNA are anti-parallel, if this is 3 prime here, this would be 5 prime. And that means this would be 3 prime. Okay, it's always opposite, right? The numbers, because of the anti-parallel property. And if I take a look at over here, the second limitation for DNA polymerase 3 is it can only build new DNA in the direction of 5 prime to 3 prime, which means when the DNA polymerase 3 comes, right, this is the DNA polymerase 3. I'm just going to label it for you. It can only go in this direction, 5 prime to 3 prime on the new strand. It's not able to move backwards and build like this A, and T. It's not going to be able to go back and do those. Okay, it can only go forward. Right. So as the DNA polymerase 3 moves forward, it will build more strands. Okay, I'm going to use black here because we're back to DNA. So T, A, T, C, C, and then it's just going to keep on going and going and going. And it's going to be able to follow the helicase as the helicase opens up more nucleotides, like maybe another C here, G, T, and A. Then the DNA polymerase is just going to keep on going and and continue to build the new strands. Let's take a look at the other strand and see if we're going to have a problem. Okay, so you're going to have another primase, and then you're going to have another primer here. So for example, you will have GAU, AU like that. Okay, that's going to be another primer. Now look, this is phi prime, which means over here it's going to be an opposite number. So that's 3 prime, and that is phi prime. Okay. And when the polymerase 3 comes, it's going to go this way. Right? It's going to build in the phi prime to 3 prime direction on the new strand. There is a CCG here, sorry, GGC here. So the new one would be CCG, A, and T. And then it's just going to keep on going, no problem. But what about these stuff here? What about all these nucleotides? How are we going to copy those? Well, it turns out as you open up the fork more and more, in order to, the DNA polymerase 3 cannot move backwards. So in order to make copies of these DNA, you would need to have another primer. So a primase would come and then it would lay down a few more nucleotides. Okay, and then the polymerase 3 will come, another polymerase 3 is going to go build this way. And as you expose more and more nucleotides, well, guess what? You're going to have another primer. And then it's going to go like that. Okay. Any questions before I tell you even more? I know it's already very confusing and overwhelming, but anything you want to ask at all? Oops. Now, this strand here is being built in one go with no breaks. And this one is actually being built in several stages, right? In several fragments. So we give them different names. But I just want to redraw this for you in a clean diagram to help you get a better picture of it. Okay? So maybe you can follow along. You can draw your own as well. So we have the replication bubble. Like that. And this is 5 prime, and this is 3 prime. This is 3 prime. This is 5 prime. We were just looking at half of the bubble, so the left replication fork. But remember, the whole bubble is for both sides, right? Now, we already know that there is a helicase here that would unzip more on one side, and then we will have another helicase here that will unzip more on the other side. To copy the DNA, we need to have the primer. So I'm going to use pink just like before to draw the primer. I'm not writing the ACTGs to keep it simple. Let's zoom in a little bit here. Okay, I want you to understand the numbering system. What number should I put over here for this primer on the right side here? Should I put 5 prime or 3 prime on the right side over here? 5 is correct. If you follow this strand, the black one is 3 here. And on the other strand, it must be an opposite number. So that's 5 prime and 3 prime, which means when the DNA polymerase 3 comes, it's going to build in this way. So nothing is new. It's exactly what we did in the previous diagram, but just without the ACTGs. But I want you to understand on the other side of the bubble, which we didn't draw, you would have to put another primer here and another primer. And the polymerase 3 will continue to build in this direction like so. Okay. So for the side that's being built continuously with no breaks, That is called the leading strand. Leading strand. It is built continuously with no gaps. But on the other side, these are being built discontinuously. There are multiple pieces. So we call it the lagging strand. So it's built discontinuously in fragments. On the flip side of the DNA, of the bubble rather, things are the other way around. You're going to have a primer here, and then the DNA polymerase 3 will be able to grow over here. Okay. So this becomes the leading strand. And I'm just going to add more details here. Leading strand grows into the fork, the replication fork. Right? It just follows the zipper, in other words. But on this side, since the polymerase 3 cannot move backwards, you're going to need to have more primers and more primers. And every time you add a primer, you will need to build the DNA with the polymerase 3. And it's being built in fragments. So these are the lagging strand, which grows or builds, doesn't matter, grows away from the fork. So if you look at one side of the fork only, one side, then one strand is leading and then the other is lagging. And then on the other side of the fork, it's also one strand being leading, the other being lagging, but the position has been switched. Now these little fragments, the DNA fragments that are being built a little bit at a time, they are called Okazaki fragment, named after the Japanese scientists who discovered them, Okazaki fragments. Okay. So over here, also Okazaki fragments. So that's part of the story so far. We're not going to be able to finish it. I mean, I could, but then it's probably too much information already. So we'll resume next class, and I'll tell you what happens with these primers, because the primers are RNA, and we need to actually remove them to finish the process, right? And what I would ask you to do is to organize your notes a little bit, maybe redraw some of these diagrams and try to remember steps up to this point. And on Wednesday, we will continue with the notes. We'll add more details to it. Okay, that's it for now. Yes, question, go ahead.
Can I have a quick word with you when everyone leaves, please?
Sure, yeah. Okay, that's fine.
Thanks.
Okay, if you don't have other questions, you're free to go. I will see you all on Wednesday. Thank you.